I don't know how this recording is going to go on. So good for everyone who's here. All right. So we left off talking about signal transduction, and just as a little side note to say, molecules that are not proteins that take part in the signal transduction pathway are called, yay, they're called second messengers. I can write, it's beautiful. That was very scary. Okay, but proteins that typically are going to show up in the signal transduction pathways are kinases are a big one, and then also phosphatases. So if it's a bunch of kinase proteins kind of phosphorylating each other in succession, we call it a phosphorylation cascade. And what turns off kinases and what turns off phosphorylation cascades are these proteins called phosphatases. And phosphatases do the opposite of kinases. They remove phosphate groups from proteins. So adding and removing phosphates can go in either direction in terms of activation of proteins. Adding a phosphate can activate proteins, and removing a phosphate can also activate proteins. So this is showing you just an example of a specific signal transduction pathway that happens with signaling from the ligand epinephrine. So let's kind of look at what happens. So epinephrine comes in, binds to a GPCR receptor. When the GPCR becomes activated, that causes the G protein to convert its GDP into GTP. That causes the G protein to be active, and it can go and activate other proteins. In this case, it's activating adenylyl cyclase. So adenylyl cyclase is going to convert ATP into cyclic AMP. Cyclic AMP is going to activate protein kinase A, which then activates another kinase, which then activates glycogen phosphorylase. And glycogen phosphorylase converts glycogen into glucose. So we've activated a bunch of things in succession and caused a cell response at the very end. And the idea of this signaling pathway is if epinephrine is around, release glucose stores. So when does that happen? Epinephrine is a signaling molecule, a ligand, that gets released during your fight or flight response. So the idea is fight or flight is going to signal to your cells to release a lot of glucose. Why? Because glucose is used for cellular respiration. Cellular respiration takes glucose and makes a ton of ATP. So it's a way to make a lot of energy for your cells so that they can all activate at once and hopefully help you escape or fight some kind of threat. Right, this is showing you an example of a phosphorylation cascade. Signal binds to a receptor, activates some protein, that activates some kinase, that activates another kinase, that activates another kinase, that causes, in this case, a cell response that involves activating a transcription factor. So call back to gene expression unit. So the activated transcription factor will bind to a gene, and now you know, you're smart and you're like, oh, that's a control element of maybe a proximal control element or maybe an enhancer, and that causes increased transcription, and you'll create, in the end, some kind of protein. So remember, the key with transduction, the reason why we don't just activate one kinase that goes and causes the cell response, is that at each step in this transduction pathway, we get amplification, right? So maybe we activate 10 relay proteins, and each of those 10 activate 100 kinase 1 proteins, and then we activate 1,000 kinase 2, 10,000 kinase 3, and so we can activate way more transcription factors in the end because we have all these different steps. Okay, what is a relay molecule? It's just a molecule that is one of the proteins or molecules in the signal transduction pathway. So they kind of act like in a relay, right? Activating each other. When a protein is activated, what happens to it? You should know that when a protein's activated, it changes shape or changes conformation. What is a phosphorylation cascade? We already said number 4. What are second messengers? We already said that. What are some key examples of second messengers? So the second messengers you should know and recognize are cyclic AMP. Calcium is often acting like a signaling molecule, and it's definitely not a protein. Calcium is an ion, so we'd call it a second messenger. Other ones you might see are PIP2 and DAG. What's the end result of signal transduction? You get some kind of cell response that is amplified. What are the types of responses in cells? There's basically 2 types of responses. Either in the end, you just activate a particular protein, and that's causing a cell response, or you're activating a gene. So you're either turning on gene expression and then synthesizing a new protein, or you're just activating a protein that's already there. So activate protein or turn on a gene. And then keep in mind, the opposite response is also possible. So sometimes you're inactivating a protein. Sometimes you're turning off a gene. You can also turn up and down. Okay, so let's talk a little bit more about second messengers and those last 2 that I mentioned, the PIP and DAG. So that's related to the GQ pathway. And again, you're not going to be asked to list it, but you should be familiar with it. So if you were given a diagram, you would be able to kind of remember what generally is going on here. So what generally is going on here? The GQ pathway involves a GPCR activating a G protein that then activates not adenylyl cyclase, but a molecule called phospholipase C. And phospholipase C converts the second messenger PIP2 into a DAG protein and IP3. So actually IP3 is another second messenger. What IP3 does is it goes over to the endoplasmic reticulum, the smooth endoplasmic reticulum that stores calcium, and it binds to calcium ion channels in the membrane of the smooth ER and causes them to open and release calcium. Calcium is also a second messenger that can activate other proteins that eventually cause some kind of cell response. So again, not memorizing this pathway, but just being familiar with, okay, there are these molecules that activate other molecules and cause a response. Yeah, I think that's all I want to say about that. So we could also talk about DAG. DAG also can activate kind of a downstream signaling transduction pathway. So what DAG does is it activates protein kinase C. It's just another protein kinase. That's then going to phosphorylate other proteins that activate other proteins and eventually cause a cell response. So just more cell signaling. Okay, we get cell signaling. How does cell signaling end? So the best way to stop cell signaling is stopping it at this point of the very first signal. So stopping initial signal. So that involves turning off the production of that first ligand. Can you predict how mutations in a specific signaling protein to specific signaling proteins will affect a signaling pathway? What about chemicals or drugs that are introduced that affect certain signaling molecules? So let's look at this example of that. The diagram above illustrates feedback control as exerted by the hormone thyroxine. Following surgical removal of the thyroid gland, the level of TSH in the blood will increase. So that's the one right here. Which of the following best explains this increase? So it's saying if we remove the thyroid gland, then TSH will increase. So removing the thyroid gland is going to cause less activation or less production of the hormone thyroxine, right? Because the thyroid gland is the one making thyroxine. So if we don't produce thyroxine, what are we not going to do? We're not going to inhibit the anterior pituitary. We're not going to inhibit the hypothalamus. So if we don't inhibit the anterior pituitary, words are hard, then it's going to continue to produce TSH in high amounts. So let's see which answer choice says that. Residual blood thyroxine from prior to the thyroid removal by the cells, stealing more. So this is saying thyroxine will bind to cells in the anterior pituitary and signal more TSH secretion. This one says thyroxine will remain bound to the receptors. These body cells will secrete additional hormones that stimulate the anterior pituitary to secrete TSH. Okay, thyroxine that was stored in the anterior pituitary prior to the thyroid gland removal will signal more TSH secretion, and a decrease in thyroxine levels means a loss of inhibition to the hypothalamus and anterior pituitary, leading to increased TSH secretion. So which one of those matches what the diagram is showing you? Put it in the chat. Yes, yes, yay. You guys got it. D is the one that matches the diagram. So all of them sound good, but D is the only one that matches the diagram. Okay, feedback. So feedback can be either positive or negative, and this is just kind of an example of cell signaling. And feedback mechanisms do one of two things. So either some kind of change happens to a cell or an organism, and the response is to do more of that change. So it's kind of like pressing your foot down on the gas and then pressing it harder, doing more of it. Or you can get a change and the cell response is to go against that change. So change happens and the cell responds by saying, okay, do the opposite, stop doing that. So the first one to kind of lean in and go with the change is a positive feedback loop. And the cell signaling pathway that goes against the initial change that happens is a negative feedback loop. So what's an example? Positive feedback would be like childbirth and the contractions that happen in the uterus. So the initial signal is actually pressure on the uterus from the baby. And when there's pressure on the uterus, that's going to cause a signal to the brain that then signals to a gland to secrete a hormone, oxytocin. And then oxytocin travels back to the uterus and stimulates contractions. The contractions will cause more pressure to happen on the cervix. More pressure is going to send more signals to the brain, which causes more gland releasing of oxytocin, which just keeps causing this change to happen. You get more and more and more pressure on the cervix, more and more signaling to keep doing that. And so that's how the baby comes out. So that's positive feedback. Fruit ripening is another example. As fruits become ripe, they release a gas signal called ethylene, and nearby fruits can receive that ethylene signal, and it causes those fruits to also start to ripen. And then as they start to ripen, they're going to release more ethylene. So you're getting an initial change of become more ripe, and you get more and more and more of that change. Okay, what about negative feedback? Classic example is body temperature. So you get a change, and the cell signaling pathway will work to go against that change. So your body temperature gets high, the cell signaling response is to try to get the temperature back down. So one way it does that is to cause your blood vessels to dilate or get bigger, and they can release heat that way. Other responses to your body temperature rising are like producing sweat, because sweat can cause evaporative cooling, where you have liquid on the surface of your skin, and as the water molecules heat up, the hottest ones, the ones with the most energy, will convert to the gas state, and so that will cool down the surface that the water evaporates off of. And then the opposite is also true. If your temperature goes up, then we will do if your body temperature goes down, your body will respond in ways to cause your temperature to go back up, such as vasoconstriction. So that will conserve heat in your blood. Shivering, so your muscles making little contractions, and that will cause your body to heat up. Okay, another example of negative feedback, that's like the classic example that you'll probably see on the AP exam, is the negative feedback loop of blood sugar or blood glucose levels. So as your blood glucose levels rise, your body's going to work to make them go back down. So we kind of already went over this, but as your glucose goes up, your pancreas will release the hormone insulin, and insulin will trigger a cell signaling pathway to say, take up glucose and form glycogen. If your blood glucose levels goes down, so you have low glucose in your blood, your body's going to work to release glucose into your bloodstream. So again, it's your pancreas, but now it's going to release the hormone glucagon, and glucagon will signal to the liver to break down glycogen and release glucose molecules. So again, you probably won't have to memorize particular feedback loops, but you'll be given diagrams of them, and you should be able to recognize what's happening in the diagram, and having some familiarity with examples will help you with that. All right. So that is cell signaling, and we can move on to cell cycle. So cell cycle, again, refers to the steps that a cell goes through in order to divide. So what is the point of a cell dividing? Why does a cell do that? The reason a cell divides is to grow, so organisms getting bigger. It might be for repair. So maybe the existing cells have become old or damaged and they need to be replaced. Yeah, so those are kind of the main reasons cells will divide themselves. I guess the last one would be some cells, that's just their main form of reproduction, right? Like bacteria cells, they divide and create two identical copies, and that's just how they reproduce themselves and make their population continue. And that's called binary fission. So hopefully right now, there's some binary fission happening in our classroom so that we have bacteria to look at tomorrow. Okay, so more than memorizing the steps of the cell cycle, what I am sure you're going to be tested on is understanding the regulation of the cell cycle. So the cell signaling that happens to get a cell to go through the cycle or to tell a cell to stop going through the cell cycle, and how messing with that can cause cancer. Okay, so briefly, what are the stages of the cell cycle? You should know there's G1, S, G2, and M. G1, S, and G2 all make up what's called interphase. And then there is the M phase, which is the mitosis phase. And what you should remember is that the two really main parts of this are the S phase and the M phase. So the M phase is the actual physical division of the cell, and the S phase is super extremely important because the S phase is when we do DNA replication. So when we talk about DNA replication, you guys know all the steps and all the enzymes. You have to connect that process to the S phase. That's all happening in the S phase because it would not be good to divide a cell with only one copy of DNA. You need to have two copies of DNA to make two cells. So remember the S phase and how important it is to making more cells, please. Those two are the main phases. The other two phases, not super important. They're for growth and prepping for those other two phases. Other big things to know about the cell cycle. Notice how most of the time when a cell is actively in this cycle and trying to divide, it's actually an interphase most of the time. So most cells, typical cells, can divide every 24 hours. 23 of those hours are spent in interphase, and 1 hour is actually spent in the M phase. So most of the time when cells are dividing, they're not physically dividing, they're in one of those pre-stages. Okay, so again, G1, mostly think growth and prepping for the S phase. G2, think growth and prepping for the M phase. And then there's that little side stage that happens that's really not a part of the cell cycle, and we call it G0. And G0 is when the cell leaves the cell cycle. It's no longer trying to divide, either because it's not appropriate to divide right now, we don't need more cells, or because the cell is specializing, or it's becoming differentiated. So cells that take on a specialized function, like certain types, well, all the different types of skin cells, neurons, muscles, once they become differentiated, they're in G0. They're not going to divide. So it's really like skin stem cells and neuron stem cells that are the ones actively dividing. But once they become specialized, they're not going to divide anymore, and they go into G0. Okay. So one thing to note about the cell cycle is remembering the importance of cyclin molecules and also CDKs to controlling how the cell moves through these phases of the cell cycle. So remember, CDKs are proteins. They're proteins called cyclin-dependent kinases because they're inactive by themselves, but once they have a cyclin molecule bound, they become active. And what do they do if they're a kinase? They phosphorylate things. And typically when they phosphorylate things, they say, go to next phase. And how this works with the cell cycle is there's a bunch of different CDKs and a bunch of different cyclins, but the CDK concentration throughout the cell cycle stays the same. However, the cyclin concentration of different types of cyclins changes throughout the cell cycle. So what this means is it's really the cyclin's changing concentration that controls the movement through the cell cycle. So you can think there's a particular cyclin that rises in concentration between G1 and S. So when it rises in concentration, it's going to bind to a CDK, and that CDK is going to activate a cell signaling pathway that says move on to S. And what concretely happens in that process is a cell response that makes all the proteins or activates the proteins needed for DNA replication. And then there's going to be another cyclin that rises in concentration between S and G2 and activates a CDK that causes a cell response that says move on from the S and go to G2. And there's another cyclin in CDK that rises in concentration at the end of G2 and says go from G2 to M. But importantly, it's the cyclin molecules that change in concentration and the CDK stay constant. All right, what are the phases of mitosis? You're not going to have to know the details of these phases. You should just know kind of briefly what happens in each one of these. So prophase is the starting phase, and there's something that is significant that happens in prophase that kind of signals to you that the cell is now in the M phase and no longer in interphase. And it has to do with how condensed the DNA is. So DNA condensation is the key to prophase. So throughout interphase, what does the cell's DNA look like? It looks like spaghetti. It's all kind of out and not condensed. And in that form, it's actually chromatin. But once we go to the M phase and we enter prophase, we're now going to condense the DNA up into these very tightly packed pieces of DNA called chromosomes. It's the only time you see DNA as chromosomes. And the reason why we do that is because it's way easier to divide those than it is to divide that tangled mess. But we need it to be a tangled mess most of the time because that's how we can actually access the DNA and read it. So prophase, think DNA condensation. Prometaphase, key things that happen is we get the kinetochore proteins that will attach to the center of those chromosomes, which are called the centromere. Remember, the kinetochore proteins are important because that's how these spindle fibers that will eventually pull the chromosomes apart can actually attach to the chromosomes. They attach at the kinetochore. Okay, so condense DNA, get our kinetochores. Metaphase, M, middle, all of the chromosomes line up in the middle of the cell, not magically, but because they have spindle fibers attached on either side, because they have those nice kinetochores. And on both sides, those spindle fibers that are made of microtubules are trying to shorten and pull in both directions. So if you're getting equal pulling on both sides, you just end up in the middle. That's how we get metaphase. Anaphase, what happens is the chromosomes get pulled apart. So initially we have those chromosomes sticking together because of cohesion proteins. In anaphase, we activate cell signaling pathways to get rid of cohesion. And so the two sides of the chromosome can pull apart. So just as a reminder, each side is called a sister chromatid, but once they get pulled apart, we consider each chromatid its own separate chromosome. So it's only a chromatid when they are connected. Okay, anaphase apart. Telophase is easy to recognize because it's just the very last phase before we actually separate the cells. There's two new nuclei, and then cytokinesis literally means division of the cytoplasm, and it's the physical division of the rest of the cell. So anchor points are: start it by condensing the chromosomes. Metaphase and anaphase are easy to recognize, so is telophase, and cytokinesis is just the physical separation. So all of this happens really quickly within just an hour. Okay, what's the difference between cytokinesis in plants and animal cells? In animal cells, there's no cell wall. In plant cells, there is. That causes the difference. So animal cells, they form almost like a little microtubule drawstring with actin molecules that can basically tighten and pulled inward on the membrane, and that's going to separate the cells. And that point where it gets pulled in is called the cleavage furrow. In plant cells, we can't just pull it in because of the cell wall. So instead, we just start to build new cell wall parts. So there's like vesicles with cell wall parts that go to the center and start forming a new cell wall at an area called the cell plate. And then once it's fully formed, yay, we have 2 new cells. Why do cells divide? Growth, repair, reproduction. Already said that. What are cyclins and cyclin-dependent kinases? Already said that. What are tumor suppressors and proto-oncogenes? Describe how a mutation in either could lead to cancer. So tumor suppressors, you should think that's the stop, no-go signal. Proto-oncogenes, they're the go signal. They stimulate the cell cycle. They say move on to the next phase. And both are important for regulating the cell cycle and preventing cancer. So how could these cause cancer if they get mutated? If you mutate a proto-oncogene, if you mutate the go signal, what you get is saying go too much. So proto-oncogenes that are always active, or they're no longer just active when they get phosphorylated or something. They signal go too much and you get the cell dividing too much. That can cause a tumor and cause cancer. You can also mutate tumor suppressors and cause cancer. So that would look like not enough stop signal. So if this is go too much, the mutation for tumor suppressors is not enough stop. You're not saying stop when you should be. So the cell is going to continue to go through the cell cycle too much when it should be stopping. So you can think of proto-oncogenes like pressing the gas, and tumor suppressors like the brake on the cell cycle. What is apoptosis? What regulating molecule signals apoptosis during the cell cycle, and why would this happen? Apoptosis is programmed cell death. Emphasis on programmed. It's not accidental cell death. It's when you do a cell signaling pathway to cause a cell to die. And one kind of classic signaling molecule that's related to sometimes causing apoptosis is the molecule p53. So p53 activates a bunch of cell signaling pathways that do these steps in the cell that cause it to die. So one thing it does is signal chromatin condensation. It signals the membrane to start to kind of move in and out, which is called blebbing. And then we start to break off the little pieces of the membrane that's blebbing, and eventually just continuously break down the cell more and more and more, and then recycle its useful parts. Okay. Let's see. Last thing I wanted to do, I'm probably going to go through these FRQs in class, unless you guys want to do them right now, just to not keep you guys on too long.
No, class is fine. Class is fine.
Okay. All right. I guess I went through this stuff. Yeah. Okay. Okay. I'll be done now. We can go through the FRQs and you can apply your knowledge tomorrow. Yes. And y'all have APUSH. So good luck on APUSH and get some sleep. Sorry that my stuff messed up. Good night. if you take out like the problems, it wasn't that long. Good night. If you don't have APUSH, does that mean there's going to be like no one? So APUSH is juniors. There should be sophomores and seniors, I think, in the morning.
Miss Langit, it's Anna. I just wanted to say, like, I'm sorry that I said you go on tangents because I feel like I pressure you to go like slower or faster on the thing. Then you can explain as much as you want.
No,
Yeah.
I need the pressure.
Yeah, I'm sorry. Anyways, thank you.
No problem. Bye, everyone.